Good afternoon, my dear friends. Today I am Sipra Banerjee, Associate Professor of Banerjee University from the Department of Pharmaceutical Technology, want to describe a topic on antineoplastic agents. Antineoplastic agents means the anti-cancerous agents. So we know cancer is a very deadly disease which is spreading all over the country. So first of all, we should know what are the reasons of cancers and cancer types. So, neoplasia, that is the cancer, is the uncontrolled growth of new tissue, the product of which is known as a tumor. The word actually means a local swelling. If the cancer is localized, it is said to be benign. And if the cancer cells invade other parts of the body and set up secondary tumors, a process known as metastasis. The cancer is defined as malignant. That means uncontrolled growth of the cell. That means whenever the cell is not dying in a normal process, it is known as the cancer. That is, the cells are inflamed and invading, or cell division is going in an uncontrolled way. That is known as the cancer. So the whenever the cancerous cells are growing in different parts of the body, so they are known as the malignant tissues. So generally whenever the cells are growing in an uncontrolled way, they are forming a tumor. So when the tumor is growing, if the tumor is not spreading all over the body and they are not having any pain and not causing the problems or disturbance to the another parts of the body or the parts where the cancerous cell has grown up, so it is said to be benign. And the malignant, that is the cancerous, when the cancer cell invade the other parts of the body and the problem occurs and the part where the cancer cell is growing on. Now coming to the cancerous type. First of all, coming to the sarcoma. So that is the cancer that is growing in the connective tissue like bones, nerves, muscles, tendons, cartilage and blood vessels of the arms and legs. Next, coming to the carcinoma. That is begins in the epithelial tissues of the skin or in the tissues that lines internal organs. So internal organ like stomach, like intestine or liver, if there is an organ, that is known as the carcinoma. Now lymphoma, that is a cancer of the lymphatic system of the body involving immunocells. That means in the immunogland, that is under the arms, under the legs or on the arms or thighs, the lymph glands and lymph nodes are there. So whenever the cancer is growing in the lymph node, it is known as a lymphoma. Now coming to the leukemia, that is the blood cancer that is usually benign in the bone marrow and result in high number of abnormal blood cells. Whenever any mutagen or any unwanted substance are coming to a body, the WBCs are trying to destroy them. So as a result, the number of WBCs are increasing and as well as they are depressing the bone marrow, and this process is known as the leukemia. Now coming to causes of the cancers. So first of all, 30% cancers are caused by smoking, that is the tobacco, chewing or smoking. So that is not only the cancerous cause, another causes are there like 30% are diet related. Carcinogenic chemicals in smoke, food and the environment may cause inducing gene mutations or interfering the normal cell differentiation. That means whenever the DNA replications are going on, then the cancerous cells are growing in an uncontrolled way. So that's why mutations are occur. So that mutations whenever growing in replication and in other cells it is spreading, it is known as the mutation. So the birth of the cancer, that is the carcinogenesis, can be initiated by a chemical, usually a mutagen. The chemical which is causing the cancer is known as a mutagen, but other triggering events are also responsible for the cancer. Okay. Next, going to the now there are different viruses and bacteria as cause of cancer. Like different viruses and bacteria are also coming to our body and they are working as a mutagen. They can also change the genes of the DNA. So epistem, the bar virus is the cause of Burkitt's lymphoma and the nasopharyngeal carcinoma. Human papillomavirus are the sexually transmitted and can lead to cancer of the cervix. Herpes hepatitis B may cause 80% of the all liver cancers. HIV can cause Kaposi sarcoma and lymphoma. So in case of HIV, how can you detect it? If the person is having some sarcoma or some hairy like structure in the upper mouth portion, it is known as the Kaposi sarcoma, and that will be having whitish in color. Now, the bacterium Helicobacter pylori is the responsible for the many stomach ulcers, and it is also implicated in the stomach cancer also. Now, genetic faults leading to cancer and proto-oncogenes and oncogenes are also responsible for cancers. Whenever some genes are converted to oncogenes, that means that genes will be converted to cancerous cell. So, the proto-oncogenes are genes which normally code for proteins involved in the control of cell division and differentiation. If they are muted, they disrupts this normal function and the cell can become cancerous. So the proto-oncogene is defined as the oncogene. That means the oncogene or the oncology, that is the cancer therapy or cancerous problem, when the genes are first in the stage that is known as the proto-oncogene. Now, inactivation of the tumor suppression genes, some of the anti-oncogenes are there, that is activating the that is deactivating the oncogenes, that is the suppression of the. If the anti-oncogenes are suppressed, then it also lead to cancer. Next, coming to the cell cycle regulation, we know the G1, M, metaphase and cell cycle phases are there. So when the metaphase are going on, this is known as the cancer. Next, I'm coming to the these are the not required. I'm not going to discuss elaborately here because you have to know the drugs for the cancer. So angiogenesis, that is also tissue invasion and metastasis. Tumor cells are and causing invasion, and as a result they are causing the angiogenesis, that is they are spreading in a numerous way. Now, most important part, classification of the antineoplastic agents. So they are also classified into alkylating agents. The alkylating agents are classified into four categories: nitrogen masters, then ethylenamine, then nitrosoureas, alkyl sulfonate, triazine, and methyl hydrazine. Next, coming to the platinum coordination complex, that is the cisplatin, carboplatin, oxaloplatin. These are the very costly drugs. Now coming to the antimetabolites, that is the folate antagonist, methotrexate, that is the acting against the different folic acid synthesis, purine antagonist, then pyrimidine antagonist. These are the important drugs like 6-mercaptopurine, 6-thioguanine, azathioprine, fludarabine. These are the purine antagonist. Now coming to the pyrimidine antagonist, 5-fluorouracil, doxyfluoridine, cytarabine, gemcitabine. These are the important drugs for the pyrimidine antagonist. Now coming to the description of one by one. Now there are some cytotoxic drugs which are also causing as acting as anti-cancer agent, but they are in cytotoxic in nature. It's like microtubule damaging agents. So that means whenever in our cell the microtubules are forming, is the microtubules are damaged, they can also cause leading to the cancer. That means whenever the partition of the different cell partition is not can be formed, then the different organelle will not be formed. As a result, the cancerous cell will be forming. So natural example, zinc alkaloids, taxanes, estromastine, these are. Topoisomerase 2 inhibitors. These are the different enzymes which are responsible for the genetic replication of the DNA replication. So topoisomerase 2 inhibitor, topoisomerase 1 inhibitor, like topotecan, irinotecan. These are the antibiotics. These are the actinomycin D, doxorubicin, daunorubicin. Like these are the important bleomycins, mitomycin C. These are the important classes of antibiotic. Next, coming to the miscellaneous, which are not coming to any classification: hydroxyura, L-aspergillus. Next, arsenic trioxide. These are the different category coming under the miscellaneous category. Now containing targeted drugs. Some of the targeted drugs are there which are acting specially on the special type of cancer, like BCR ABL tyrosine kinase inhibitor. So that means they are acting specially on the tyrosine kinase enzyme. Imitanavir, dasatinib, nilotinib. Now there are some EGF receptor and inhibitors. These are the some receptor which are present in some cells. Like next, angiogenesis inhibitor. So some of the drugs are there. Now proteinase inhibitor and CD20 inhibitor. These are the drugs not so important, but you first classify the first category of the drug. These are very important and which you should write in exam. Now coming to the hormonal drugs, which are also very helpful in preventing the cancer, glucocorticoids like prednisone, estrogen, SREMs, SER down regulator, aromatase inhibitor, 5 alpha reductase inhibitor, anti-androgens, gonadotrophic regulatory hormone antigen, progestins. These are the important hormonal drugs which are also reducing the cancer. Now general toxicity, there are some toxicity of this drug like bone marrow depression. Depression of the bone marrow occurs result in the granulocytopenia, agranulocytosis. That means the granulocytes or the cells which are responsible for the blood flow or circulation, they will be decreasing in number. Thrombocytopenia, aplastic anemia, these are the various cytotoxicity. Now lymphoreticular tissues, lymphocytopenia, inhibition of the lymphocyte function. Lymphocyte will not be working properly, they will be destroyed. Now oral cavity infection and xerostomia occurs. Now GIT, like in the case of gastrointestinal tract, oral cavity infection occurs on. Next, diarrhea, shredding of mucosa, hemorrhages are there. Now skin, alopecia. Maximum cases, the cancerous cells are highly replicating in nature. That's why the cancerous drug always targeted the cell which are growing in nature. So in case of skin, in case of hair follicles, they are growing in a larger way. So that's why they are also targeting the drugs. As a result, the hair fall, beards, the eyebrow, and the mosters, these are also uh, disappear. Now gonads, in case of gonad, different ovulation, amenorrhea occurs, inhibition of the ovulation. So after the cancerous, many persons loses their fertility also. Fetus, in case of fetus, absorption occurs, fetal death occurs, teratogenesis may be there. Now carcinogenicity, hyperuricemia, these are the different type of toxicity may be occurring due to the carcinogenic drugs. The mechanism of action. So first of all, now you should know why the drugs are acting and what are the ways of acting. So in biochemistry, you have seen about the cell cycle pathway or the replication of the gene or the DNA. So you see that in case of DNA, purine and pyrimidines are there. Ribonucleotide, deoxyribonucleotides like the RNA, DNA, they are the conversion process. So from DNA to RNA and proteins, enzymes, microtubules are going on. So different drugs are working in this pathway in different way. Like antimetabolites, 6-mercaptopurine, 6-thioguanine, methotrexate. So they are inhibiting the purine synthesis to ribonucleotides. So as a result, the cancerous cell will not be growing. Now coming to the hydroxyurea, they are working on the ribonucleotides to, see here, ribonucleotides to deoxyribonucleotide conversion will be prevented by the hydroxy. So 5-fluorouracil, cytarabine, gemcitabine are working from deoxyribonucleotides to DNA conversion. Okay, so as a result, DNA will not be produced and the cell will not be invaded. The etoposide, topoisomerase 2, they are working on the topoisomerase 2 enzyme. As a result, DNA breakdowns. So DNA to RNA will not be converted. So some of the antibiotics are there, they are working on the DNA and some of the proteins. So as a result, DNA to RNA and protein will not be synthesized. Some alkylating agents are there. These are the most important, which will alter the structure and function of the DNA by cross-linking and fragmenting the DNA. So they are the working on DNA. Now some of the vinca alkaloids, they prevent the polymerization of the microtubules. As a result, cell organelle will cannot be formed properly. Now taxanes enhance the polymerization. So this type of drugs are very important. Now we will classify this drug according to their structure and mechanism of action, their side effects and uses. So this chart is very important for all the drugs mechanism. Whenever we will be discussing, we will be going for the mechanism and you should know how their drugs working. Okay. So first coming to the activation of the agents, mechanism of action. So activation and alkyl agents how they are working. Okay. Now nucleophilic attack, unstable acridine as a reading ring by the electron donor. So they are causing in the DNA, see in case of DNA, nucleophilic attack is going on. So as a result, the structure of the DNA is changed. Okay, see the nitrogen where the electron pair is there. The drugs are, this is the drug is attaching in this portion. As a result, the DNA structure is altered and the cell cannot grow in a larger way. Now meclorothamine, that is the mustrogen or nitrogen master mucin. See the structure, 2,2-dichloro-N-methyl diethylamine, Y22 at the second position of the nitrogen, 2,2-dichloro. See the chloro position, and N-methyl, nitrogen contains methyl group, diethylamine, N contains methyl group, and diethyl means CH2, CH2, CH2 ethylamine. So it is used in the Hodgkin disease in combination with vincristine, procarbazine and prednisone. Most serious toxic reaction is bone marrow depression, which results in leukopenic and thrombocytopenia. So meclorothamine, how it can be synthesized? From the ethylene dioxide, it is reacted with the methylamine. As a result, it produces this structure. The ring is opening, then is reacted with the SOCl2, thionyl chloride, thionyl fluoride, and it produces the meclorothamine, whereas OH group will be reduced with the chlorine. Now coming to the chlorambucil, that is another category of the drug that is working on the chronic lymphatic leukemia, Hodgkin lymphemia and non-Hodgkin lymphemia. Now cyclophosphamide, they are working the myeloma, chronic lymphocytic leukemia, acute leukemia of children, Hodgkin disease, breast and ovarian cancer. Cyclophosphamide is a oxygen and phosphorus containing 6-membered saturated ring. Mechanism of the cyclophosphamide, here it is showing again, that is working on as the metabolites. As a result, toxic metabolites cannot be formed. Next, busulfan, then 1,4-butene-diol dimethane sulfonate, used in the treatment of the chronic granulocytic leukemia. Then melphalan, that is the alkarin. One second. Now coming to the different classification of the melphalan, that is the 4-bis-2-chloroethyl amino N-phenylamine. So melphalan is active against multiple myeloma, breast, testicular and ovarian cancer. Now coming to thiotepa, that is the thio means sulfur, phosphorus is there, 3 nitrogens are there having a aziridine group. So tri-1-azirydyl phosphine sulfide has high toxicity and seldom used in the ovarian and bladder cancer. Now coming to the antimetabolites, this category of the drug like working against the purine antagonist, pyrimidine antagonist and folate antagonist. So for the purine antagonist, markup-to-purine, thioguanine, azathioprine are there. Pyrimidine antagonist, 5-fluorouracil, cytarabine and fluoxetine is there. Folate antagonist, methotrexate. Now mechanism of action of the purine antagonist, where they are working? They are working to the synthesis of the purine, and as a result DNA incorporation were working there. The purine is attached, purine antagonist is attached with the purine. As a result, the DNA and RNA cannot be leading for the replication. As a result, cell death occurs. The cancerous cell is dying. There is a markup-to-purine, that is known as a purine 6-thiol, that is inhibit purine nucleotide synthesis and metabolism by inhibiting an enzyme called the phosphoribosyl pyrophosphate amidotransferase, that is used in the acute monocytic leukemia. Next, synthesis of 6 markup-to-purine. In case of 7H purine 6-ol, they are converted with the phosphorus pentathiol P2S5, 6 markup-to-purine. Okay. Next, converted to 6-chloropyrimidine to 4,5-diamine. 6-chloropyrimidine 4,5-diamine is reacted with potassium thiosulfate and concentrated HCOOH. Acetic acid is there, that is converted to one byproduct and then 6 markup-to-purine. So now coming to thioguanine, that is used in the treating acute leukemia, especially in the combination with cytarabine. The adverse effects are bone marrow depression, leukopenia and thrombocytopenia, bleeding. Azathioprine, that is converted to 6 markup-to-purine and more prominent in the immunosuppression action. Primarily suppresses in the chronic myeloma, then used in the autoimmune disease, rheumatoid arthritis and ulcerative colitis. So mechanism of pyrimidine antagonists, they are working as methotrexate and working in the enzyme DHFR. So they are working on the thymidylate synthesis and preventing the DNA replication. 5-fluorouracil, that is converted into 5-fluoro-2-dioxyuridine monophosphate, which inhibit thymidylate synthesis and blocks the conversion of the deoxyuridic acid to deoxythymidylic acid. Breast, colon, pancreas, rectum, stomach cancers, it is working. Cytarabine, that is also used in the acute leukemia, chronic myelotic leukemia, meningeal leukemia, acute lympholytic leukemia, and chronic lymphocytic leukemia. Now, in case of different drugs are there, mechanism of action, DNA polymerization is breaking down. Fluoxuridine is a pyrimidine analog that acts as an inhibitor of the S phase of cell division. Fluoxuridine is the rapidly catabolized to 5-fluorouracil, which is the active form of the drug. Now, now coming to the methotrexate mechanism. The inhibition of the DHFR by methotrexate result in the deficiency in the cellular pools of the thymidylate and purine, and thus in decrease in nucleic acid synthesis. Leukemia, breast cancer, skin cancer of the head, neck, and lung cancer. Now we will follow the methotrexate synthesis. Now coming to the antibiotics, that type of class will be discussed in later on. Thank you everyone.